right, well, we'll go ahead and get things started here. Thank you, everyone, for attending our PeDRA at the virtual SID session. I am Carrie Coughlin, and along with Amy Powler, I'm welcoming you to our session. We, as this slide says, are going to start with some introductions and our keynote speaker, and then move on to our abstract presentations. And we'll have time for questions, so definitely send those through the chat. I'm going to start with just a little bit of information about PeDRA, so this Pediatric Dermatology Research Alliance, for those who may not be as familiar with this research group. And we'll make our way through the slides. And I'll get there. We have our slate of speakers. And as you can see, we have a tight schedule, but it's because there were so many good talks that we didn't want to miss any of them. In the Pediatric Dermatology Research Alliance, we have several working groups that are focused on broad issues relevant, particularly to pediatric dermatology. And the neat thing, of course, is that a lot of things relevant to pediatric dermatology are super relevant to medical adult dermatology as well as some of the procedural aspects. So we have working groups, particularly in birthmarks, focusing mostly on vascular lesions, genetics, neonatal, inflammatory. Our cancer group does reactions to cancer therapies, as well as skin tumors, as well as pigmented lesions. And then, of course, there are studies that don't fit into a box and are cross-cutting. We have a great group, and the idea behind the group is to really support current and future researchers within pediatric dermatology, clinical researchers, basic researchers, and in a collaborative way. So you can see some of our productivity to date. And at the end here, we have a few references. Of course, we are happy to have new members. We have an annual meeting that is scheduled for October. So far in person. Of course, there's always the chance to pivot to a virtual meeting. And we do have a great grant mechanism in place as well. Before actually starting our session, I want to give a big shout out to our PeDRA administrators who have totally flipped our session from in-person to virtual. And we really appreciate all of the work that went into that, as well as for our speakers who are so generous with their time and their decisions to flip their presentations to virtual as well. We really appreciate the flexibility and look forward to an excellent session. And with that, I'll pass things off to Dr. Powler.
Thanks, Carrie. Well, it's my pleasure. If we can go to the next slide, thank you. To introduce Dr. Vicki Worth. Dr. Worth is a professor of dermatology and medicine at the University of Pennsylvania and chief of the Division of Dermatology at the Philadelphia VA. She trained in medicine at Northwestern, where I first met her. She then went on to learn dermatology and immunodermatology in residencies at NYU before joining the Penn faculty. She co-founded the Medical Dermatology Society and received their Lifetime Achievement Award. She's also started the first combined internal medicine dermatology residency program in the United States. Dr. Worth co-founded and served as president of the Rheumatologic Dermatology Society. She's recognized internationally as a leader, if not the leader, in autoimmune skin diseases based on her clinical research, including measure development and validation, and her laboratory-based mechanistic studies of lupus. She's received many honors, including the Naomi Kanoff Award at this SID meeting. And I encourage you to listen to that presentation as well. We're delighted to have her address us here today on clinical developments and future directions in autoimmune connective tissue disease. Welcome, Dr. Worth.
Well, thank you very much for this opportunity to speak. And I will get my slides up here. Actually, before I do that, I have to select my slides. Okay, so here we go. Here we go. I think, can you see my slides? Hopefully. Okay. So, all right. So I really appreciate the opportunity to talk. And I think there's a lot that I've learned in the adult world that can be thought about in the pediatric world. And I would also mention that I've been to a number of the PeDRA meetings lately. And so I really appreciate the pediatric world and the derm room interactions. We'll talk a little bit about that. And I also want to mention that I have some conflicts, but I actually feel very happy about those conflicts because it means people are finally getting interested in this part of the field. And we need new therapies. So I'm going to talk some highlights in lupus and dermatomyositis today. And I want to give you, Amy already gave you a pretty good background, and it was really wonderful meeting her way back when. I won't say when, but it was a while ago. And I wanted to acknowledge the role of mentors, because mentors make such a difference. And I was at NYU, and Erwin Freeberg was there at that time, and he was more of a lab-based person, but convinced me that the lab is actually could fit nicely into some of the thoughts I had about where I wanted to go in the field. And I was funded during my fellowship by the Derm Foundation and NIH. And I was very lucky that Dr. Andrew Franks, who's an internist, dermatologist, rheumatologist, who happened to be at NYU. And at the time I was having an identity crisis about whether I was even in the right field, as the field was really going towards cosmetics and surgery. But it was wonderful to have somebody that I could work and mentor me in a lupus clinic at Bellevue. And that's, super important. I also worked in a basic complement lab. And so I think I kind of put together almost like a translational research kind of fellowship that I don't think kind of existed in the field. And I think luckily today we do have more opportunities. But I found that a great time to think about where I could see myself going in the future. Then I moved to Penn, and I must say I was pretty undifferentiated in terms of my career path. But I did join derm and then rheumatology with a secondary appointment, and I found that to be very helpful and fruitful. And then the American College of Rheumatology, which you would think is a little bit far afield from us, was a very nice home in a lot of ways. And I met people like Richard Sonheimer, Tom Provost, Jeff Callan there, and they were really great advocates for the field. And you need people to advocate and push forward a field, especially when it's really in its infancy. So one of the issues with lupus and dermato is it's really multidisciplinary. And it's important to define the role of dermatology in connective tissue disease, because some of the other fields otherwise would say, well, this is ours too, and we're going to do the skin. But we can define our patient population and what their needs are better than more people who are taking care of more of the systemic manifestations of problems. And also both of these diseases have patients who have primarily skin problems as a major component of the population. So, but it is really important to interact with other fields. I found with rheumatology in particular for what my interests are, but also pulmonary and renal. And I also have come to appreciate the need for basic translational and clinical studies within dermatology, and also to emphasize the importance of patient advocacy groups. And in particular, when you're dealing with diseases where it's more systemic disease, often these are very well, the systemic parts of the disease are very well represented within these advocacy groups. But we in dermatology need to represent the skin perspective. And I can tell you from some of the groups that I work in that I think the pediatric rheumatology perspective is very well represented. But I think there's room for more skin representation to advocate about the needs within the skin patients. And I also would say that we have to be creative about funding, because it's not something that is still as mainstream as it could be, at least in this part of the field. So I want to start with cutaneous lupus and talk about something related to criteria, which turns out to be really important if you're going to study populations. Then quality of life in terms of defining the importance of the disease, how we learn to develop measures of disease severity, effectiveness of therapies, heterogeneity, and new therapies. And so on the right, you see a very severe discoid patient and nothing's working, and how frustrating that is for the patient and trying to make that patient better. And on the left, a patient that has DLE on one cheek and acute LE on the other cheek, and trying to explain to bodies like the FDA that, you can't just do a discoid study, that 20% of our patients have more than one subtype, and it may be hard to know which subtype, but these are all dialogues and important discussions to have as one plans how a field can go forward. So many years ago, I was asked by the SLIC group to kind of comment on what was then the ACR criteria that was in the 1998 criteria for SLE. And there were 4 criteria that were dermatologic, and you only needed 4 criteria to be called SLE. And so this seemed like a lot of criteria. And so we actually, with the derm room community, and you probably recognize lots of these names, were involved with trying to indicate that maybe criteria could be improved upon in terms of how to define SLE. So that led to the SLIC criteria for SLE a full 8 years later. And anytime one gets involved in one of these efforts, you have to be prepared for it to take a very long time. But what was kind of neat about this was the fact that there were dermatologists and rheumatologists working together, and that had not happened really that much before in criteria for lupus. So you'll recognize, you know, Melissa Costner, who at the time was at UT Southwestern, and then Jeff Callan and Andy Franks. So a number of people in the derm world were involved with this effort. And you know, I would say that in itself was really a worthwhile, we figured out some criteria, then we recruited patients and tested some of those criteria. And so I think that it was a really good experience. Unfortunately, they snuck alopecia into the criteria. And, you know, you do get into points where you try to explain that alopecia is really hard to assess. And, you know, that's when you can have really interesting dialogues and understand perspectives of other fields. So that was very important. So at the end of the day, we ended up being able to compare, for instance, DLE patients who either had localized disease or generalized DLE, looking at the ACR criteria and SID criteria and trying to figure out what the differences were in the numbers of patients that were called SLE based on these two sets of criteria. And blue are the patients who had DLE with SLE, and you can appreciate that more generalized DLE patients had SLE than those with localized DLE. And although there were some mild differences between SLIC criteria and ACR overall in the distribution of, you know, those who had SLE versus not having SLE, it wasn't that, you know, big of an issue. And I would say, though, that what we were able to do with many of our studies is to use our prospective database, which now is like 450 cutaneous lupus patients, to be able to ask these kinds of questions. And that was also has been very fruitful. So, but I wanted to emphasize that rather than focusing on, in the case of lupus, on criteria, which keep changing every 5 minutes, I think it's nice to think about, you know, more of a spectrum disease when you're dealing with something which can end up at systemic and start out with skin only. And, you know, each of these subsets can be, you know, more associated with skin only, or in the case of generalized DLE and SCLE, maybe more systemic disease, and acute LE is certainly more systemic disease, but it's really a spectrum disease. And trying to slice and dice it as much as we've tried may not be that helpful for us or for our patients. And it's now even more complicated because we now have the 2019 ACR criteria for SLE. And the criteria themselves are complicated, but now you can't have SLE unless you have an ANA. And that, of course, eliminates about 7.4% of our patients, at least in our database, from having SLE who in the past were thought to have SLE, including patients with inflammatory arthritis, serositis, and we've certainly seen very sick acute LE patients with negative ANAs. Part of the motivation for these kinds of criteria was because all the SLE trials were failing, and they decided that the problem was the way we defined SLE, and so we need a homogeneous group of people to put into SLE trials. But actually, one can do that by just having entry criteria to define whatever subset you want to study, and that actually, I think, redoing all these criteria over and over again is an interesting process. But there is actually a lot of disagreement in the rheum community about yet another set of criteria, and I think, indeed, we should think about lupus as a spectrum disease. So I did want to highlight one study that Lisa Argan, who's very involved with PeDRA, did relating between PeDRA and CARA. And this is the kind of collaboration that I think can be incredibly fruitful going forward. And I've certainly found all my interactions with the ACR and with all the different lupus groups has been very helpful. And what she found in sending out a questionnaire was that there were differences between pediatric derm and pediatric rheum in terms of certain aspects of workup, risk factors that might be leading to higher surveillance, and then second and third line agents for skin disease. And some of these kinds of differences undoubtedly relate to the fact that they're seeing a different cut of patients very often than what we see. And also the fact that we may see more mild patients, but in fact, after one is in practice for a while and sees the patient with localized DLE suddenly get lupus nephritis, you get a little humbled and do think actually that doing maybe not a urine protein to creatinine ratio, but at least a urinalysis is important. But it's really helpful to do these kinds of things, to have a dialogue, to see where the differences are, and then try to figure out why we think they're differences. And I'll highlight this a little bit later as well. So those kinds of dialogues are important to have and important to continue and can lead to very fruitful studies. So one of the studies we did was to look at how the impact of lupus on patients in terms of quality of life. And we used the Skindex, which has 3 subscores, including symptoms, emotions, and functioning. And you can see the blue for lupus is one of the higher scores, which means their quality of life is really bad. And it's particularly in the area of emotions, which given what I showed you before is not that surprising. But defining this is important because very often, you know, cutaneous lupus gets written off. Oh, it's just skin, who cares? And it's really important to the patients, and we have to demonstrate that. So CLE has high unmet therapeutic need, and there's not much evidence-based therapy for CLE. Most therapeutic trials in lupus have been for SLE. CLE patients have been excluded from most lupus trials, and there have been huge numbers of trials. And that would mean if they didn't meet SLE criteria. And CLE, with or without SLE, the skin part of this is probably pretty similar in terms of pathophysiology. If it's DLE in an SLE patient versus DLE, you know, without SLE, depending on how you define SLE, it probably doesn't matter. But to do mechanistic studies and therapeutic trials of CLE, we needed to develop and validate a reliable, reproducible tool to assess disease severity. And we developed the CLASI, and you can see that was in 2005. And Jorg Albrecht was a fellow with me at the time. And it's 15 years later, and we're still working on this instrument. So this is not something you undertake very lightly. And we've had to do a lot of studies, including reliability, responsiveness, correlating with quality of life, correlating with biomarkers. And now a really fun time when we're looking at this in the context of phase 3 international trials and trying to do further validation to figure out, you know, what's meaningful to the patient, what's a meaningful improvement, and so on. But it's beginning to bear fruit, and people are getting more interested in looking at the skin in the context of the SLE trials, and even I'll show you in the context of CLE trials. So one of the validation studies was to look at responders defined as a certain change in the CLASI. And what you can see here is that they got better enough to be called a responder. The quality of life actually improved. And you can see here that this is before treatment, after treatment, and it goes down. And so they were doing better. If there was no response in the CLASI, then you can see there was no change. And I will point out that this is looking at immunosuppressive effects on quality of life in patients who had lupus, and to show you that about half the patients did not respond to immunosuppressants, which leads into another topic in a minute. So with these validated disease severity tools, we were able to begin to do more clinical and translational studies and look at the heterogeneity of responses and perhaps get some insights into more about pathogenesis and mechanistic subtypes. And as new candidate target treatments are identified, we have opportunities to study pathophysiology and clinical effects. So in our prospective database, we found that about 55% of patients respond to hydroxychloroquine of the people who need systemic therapy. And obviously that leaves almost half the patients who are not getting better with hydroxychloroquine. So we did know that from basically word of mouth, because there wasn't much literature, that quinacrine seemed to help people who didn't respond to hydroxychloroquine. And so we would add quinacrine when hydroxychloroquine didn't work. And what this shows is about 2/3 of the patients became really rapid responders within a couple of months. Their scores were going down. Even the non-responders were trending down a bit. And so this really is suggesting that quinacrine is quite helpful in patients who don't respond to hydroxychloroquine. And really beyond the topic of this talk, but quinacrine right now has gone away for a lot of different reasons. But it's still helpful to think about subsets of these patients in terms of why there are differences in response. So we know there are PDCs, and this is showing you with CD123 is labeling the PDCs, these brown stain, that are present in the skin of a lupus patient. And also BDCA2 is another marker for PDCs. And PDCs, RNA and DNA can activate them through TOL7 and TOL9. And in CLE, PDCs, among other cell types, make type 1 interference and thereby amplify lesional inflammation. And they regulate interferon-inducible chemokines and recruit T cells into the skin. So they're thought to be an important player in lupus. And we're interested in what happens in the peripheral blood in patients who have evidence of lupus activity by the CLASI. And so what this is showing you is that many patients who have either SCLE or DLE have elevated interferon scores. So they're genes that are upregulated by type 1 interferons. And you can see that this is in the blood even when they don't have systemic disease. And in fact, the interferon score is correlating with their amount of skin activity. So we were interested in trying to understand, again, why are there some people that respond to hydroxychloroquine and some that need quinacrine? And so we looked at the PDCs and found really no difference between these two groups. But then we looked at myeloid disease, and what you can see here is there are many more myeloids in the skin in many of the patients who needed quinacrine versus those who needed only hydroxychloroquine. And what we did then was we actually did simulated PBMCs in culture, and we're able to show that hydroxychloroquine and quinaquine can block interferon, but not TNF. Hydroxychloroquine doesn't block TNF production, but quinaquine does. So then here we went on to look at myeloid DCs and also monocytes. And again, you can see LPS stimulated cells, in particular here, myeloid DCs, you don't inhibit TNF production when you add hydroxychloroquine, but quinaquine you do. But trying to understand why there might be differential effects is important. And we found the same thing, by the way, with monocytes. So no effect in decreasing TNF when you added hydroxychloroquine, but a big change with quinaquine. So then we went back to the tissue of treatment-naive lupus scan, and we were able, as you know, to follow with our database prospectively over time to determine who was a responder and who wasn't. And then we could go back to the original biopsies and figure out what was different about their gene signatures in the skin. And so we looked at type 1 interferon signatures in hydroxychloroquine versus hydroxychloroquine quinaquine responders. And what this is showing is that the hydroxychloroquine responders had higher type 1 interferon gene upregulation, such as LY6E, OAS1, MX1, but really across the board, there was a definite difference between those who responded to hydroxychloroquine versus those who needed quinaquine. But when you look at TNF, it's just the reverse. So with the hydroxychloroquine responders, there was not that much of a TNF mRNA upregulation, but it was quite different with the patients who needed quinaquine, suggesting that there are some really basic differences in the cell types and the cytokine signatures that are driving the response to therapy. So the incidence of refractoriness is a big issue. And we know that, as I said, about half the patients respond to hydroxychloroquine, another 25% respond to immunosuppressives, but there's another 25% that really have refractory disease of the patients who need systemic therapy. And so this was looking at including thalidomide that probably 10% don't respond to anything, but thalidomide has lots of issues. We were interested in figuring out about this refractory group, and what you can see is that they're much more in the way of smokers in refractory versus non-refractory. And so I like to use this data to try to convince my patients to stop smoking, and I've actually been having more success lately. So we were interested in thalidomide analogs because one of the problems with thalidomide, although it works, is that it actually and these are more potent, potentially less neurotoxic, but haven't been used as much. And so one of those would be, for instance, thalidomide derivative lenalidomide. So we did do an open label trial with lenalidomide, and this is looking at baseline and then at week 6. And you can see that the Redden scale here is dramatically better already in 6 weeks, and there's much less lymphocyte infiltrate at week 6 than at the beginning. And this is actually that patient where we had, by protocol, given 5 milligrams of lenalidomide. It came down in terms of the skin activity. And then we backed off to every other day. And you can see she got worse. And then we went back to every day and she got better. So this was a nice N of 1 sort of indication that at least for some patients, this can help. So how did our 5 patients do? Well, 4 out of the 5 got better, as defined by a significant improvement in CLASI. 1 out of 5 did not improve their skin, and 1 out of 4 of the 4 that improved had new onset proteinuria and arthralgias, which was a little upsetting to us, and we had to take her out of the study. So clearly we felt there was a need for more systematic study before we would implement lenalidomide. We did actually have funding from the Lupus Research Alliance, and we're able to look at the fact that there were activated T cells in these patients who responded, and in fact also the patient who got arthralgias and proteinuria had an increase in her activated PDCs in the setting of getting lenalidomide. So that was also a bit concerning. But then there was a Spanish study with 15 patients where there was 86% complete response without seeing any particular problems of flares. And you can see a very rapid drop in the CLASI score. Really, after 4 weeks, these patients were doing much better. And so I think many of us now are using lenalidomide preferentially to thalidomide, and I would have really loved to have seen lenalidomide developed for cutaneous lupus, but it's an oncology drug. The cost is exorbitant. It's like $650 a pill. And so they weren't interested in developing it for an inflammatory indication, which can be an issue. So they developed a new drug called CC220, which is another derivative. And these all seem to work on Ikaros and Aiolos, which are zinc finger transcription factors involved in immune cell development. And the mRNA for these zinc finger transcription factors is overexpressed in SLE. and there are polymorphisms of them that have been associated with increased risk of getting SLE. So CC220 causes a reduction in these and has effects on B cells, T cells, and monocytes. And this drug was screened to be more potent than thalidomide or lenalidomide. So they did a phase 2A study, which was basically read as placebo. You can see the skin didn't change, but the 4 treatment arms doses did trend down in pretty significant ways. And so they went on to do a phase 2 trial, which has not been reported out yet. And it'll be very interesting, but again, this was a good indication of why dermatology has to be involved in these kind of studies, because although this study really, and we know that thalidomide and lenalidomide are mostly skin drugs, they finally developed a pathway with their phase 2 that was really focused more on SLE. And those patients have more antiphospholipids, more tendency to get clots, and it became very complicated. And so the question is, can we redirect some of the development pathway towards the skin? So there are other new treatments coming out now, such as anifrolumab. There was a phase 3 trial, actually 2 phase 3 trials and a phase 2 that was successful, and one of the phase 3s was successful. And so the idea is going to be that this may well end up getting approval, and I'll show you some data, and it probably works in skin pretty well. I mean, the data suggests that. The problem is these SLE trials, as we saw with Benlysta, they get approved for SLE, and it's very hard to get it for CLE patients. And so there are a lot of regulatory issues about SLE trials when they're doing skin as a secondary outcome in terms of whether we will be able to access those drugs. There was a ustekinumab phase 2 trial that was successful, and they're now in a phase 3, and there are lots and lots of approaches, and this is such an exciting time. So this is showing you basically what's going on in a phase 2 anifrolumab trial. So this is an antibody against the IFNOR receptor, so it's blocking all the type 1 interferons. And you can see that this is a placebo effect, and the 2 treatment arms, actually, this is a percentage of responders. So it's much better response than the placebo. This is before and after. One of the caveats is that a lot of these trials, they don't really very well characterize the type of skin disease. So even if they get better, you don't actually know what the subtype is that got better. And that's another problem that, again, has required lots of dialogues and trying to influence some of the thinking there. But this is also potentially a very interesting agent. Now, this was ustekinumab, and this was presented at the ACR last fall, and essentially when we used to have live meetings. And so essentially what this is showing is that the skin got better. So this was the treatment arm, and this is the placebo arm, and there was a difference between them. We always know there's a placebo effect, but this is showing partial improvement, which can be quite important to patients. And this was another outcome measure that did not detect that change. We can see here that there was no difference in complete responders between placebo and ustekinumab. So it really is important to be able to capture what's a meaningful partial improvement. But these trials also provide an ability to actually learn more about the instrument. And so what they did, and what we've done, is to look at different baseline scores of the activity in the skin and be able to show that even down to low, mild disease activity, a 35% improvement was able to show a difference between the placebo arm here in gray and the treatment arm in purple. So then you come down to, well, is this significant disease for the patient? And so there are a lot of issues about, when you start thinking about what's important to a patient and what's a meaningful improvement. But clearly this is able to show differences at multiple different entry levels of disease activity and different cut points for improvement. So that brings me to another drug, which is very interesting. And I told you about BDCA2 as being a marker of PDCs. It's a receptor on the PDC membrane, and it gets internalized when this antibody binds. And when that happens, you get a decrease in type 1 interferons, pro-inflammatory chemokines, and also pro-inflammatory cytokines. So this was a phase 0 trial that we published last year in the Journal of Clinical Investigation. And what it's showing is different subsets that were treated. This is day 0, before treatment and after. And this is after 1 injection. You can appreciate that the purple, which is actually mix A, which is again upregulated by type 1 interferon, goes away in the responders, and so does the score in the skin. And so there were obviously non-responders that I find particularly interesting to understand what's different about them. But in any event, this is a drug that looks very interesting, and the phase 2 just is getting read out. I'm presenting it next month at the EULAR meeting. And what was very neat about this trial is that they did SLE and CLE. So there's an arm that's CLE and an arm that's SLE. And in the CLE arm, you can appreciate that the 3 different drug doses, like 40% of the patients got better relative to placebo, which is much lower. So this is really interesting and good news. And we actually treated 3 of the patients at our site, and they stayed in remission after their final injection, which was monthly sub-Q, for about 5 months. So, you know, it really does seem like this will be very interesting to take into a phase 3 trial. So in summary, cutaneous lupus impairs quality of life and has high therapeutic need. There are different cell subsets and cytokine signatures associated with the heterogeneity of response to therapy. And PDCs play a critical role in disease in many patients, and there are increased myeloids that correlate with the response to quinacrine. There are basic insights that have led to successes in new therapies for CLA targeting interferon, and there's really a need for personalized medicine in order to continue to, and better understanding of the disease heterogeneity in order to target the right therapies. So in my last 5 minutes, I'm going to talk briefly about dermatomyositis. This is typical dermato with, erythema over the joints and Shaw sign. We know the interferon signature is more beta and gamma rather than alpha. So there's something different about these patients. And we know the criteria, back to criteria, because again, if you have to know who has dermato. But if you look at this is from 1975, and for something like 45 years, these criteria really stuck. And so essentially, you had to have, if you had a rash of dermato and one criterion, you could be called possible DM. But look at the criteria, they're all muscle criteria. So these patients couldn't get diagnosed with amyopathic DM, and it was a huge problem. And Rick Sonheimer really, tried very hard to get amyopathic DM understood. So what we noticed in our practice is that, and we now have about 350 dermato patients, but we had 232 confirmed cases, and about 44% of them came in the door with the diagnosis of DM, but the others have been called SLE, undifferentiated connective tissue disease, undiagnosed, or other. So these patients truly have a hard time getting it diagnosed, and they have ILD and so on. So it's a problem. So we now have new criteria. Ingrid Lundberg and I work very closely with the steering committee to develop new criteria, which now include 3 skin criteria. And if you have 2 of them, you can be called likely dermato. This is very helpful for our patients. But it works for only about 74% of our patients. So these are classification, not diagnostic criteria, but we think we can do better. And one of my fellows, Dr. Joseph Concha, is now working on an effort to try to improve the skin criteria. And we're working very closely with the rheumatologists and who are experts in this field as well. We have a CDASI, which is similar to CLASI, and all the studies that had to go along with that to try to validate. And we were interested in using that to look at many aspects of activity. This is just to show you the kind of translational studies you can do. We looked at IL-31 and lesional DM versus non-lesional in healthies. And this is looking at the mRNA expression of IL-31, and clearly higher in lesional DM and correlating pretty nicely with itch. But there's a lot more work to be done on itch and DM. We also found many more myeloids in skin, more like our quinacrine patients than, and very few PDCs, which is also interesting. And we found, although maybe a quarter of the patients respond to hydroxychloroquine that have dermato, the non-responders also tend to have more myeloid DCs. So, and no big differences in the PDCs. We've been able to show that interferon beta is produced, and this was actually work that Jay Patel and Kristen Chen have been working on, looking at interferon beta produced by CD11C cells, and so they are capable of making interferon beta. And Spandana Mataruki has been working on flow along with Kristen Chen, who will be a derm resident in Madison. And essentially what you can see is the myeloids don't make interferon beta, the myeloid DCs, if they're from healthy controls. But if you look at dermato patients, they do, and these are the cells that are in the skin. So we do think that myeloids are an important cell type for dermato. We've gone on to do more CyTOF work, and some of it's reported at this meeting, to look at pathways in cells that seem to be important in DM. And so again, just some of the examples of translational studies one can do in these diseases. And I want to end with the lenabasum study that we've been working on now for a number of years. It's a non-psychoactive cannabinoid that binds to the CB2 receptor on immune cells and downregulates pro-inflammatory cytokines thought to be important in dermato. And here you can see in culture, we stimulate PBMCs, and then we add lenabasum in a dose response kind of way, and interferon alpha and interferon beta come down when we add lenabasum, this cannabinoid derivative, to stimulated cells. So we were able to get an NIH grant to help us with a placebo-controlled randomized trial with 22 patients, which we did this placebo trial. was a difference between the arms, and also we looked at the number of patient-reported outcomes. So here you can see the placebo versus the treatment arm, and there's a difference. And this has really been fun to show, and emphasizes the importance of skin and being able to do a short study to show impact. And this is the kind of change that we saw in patients who responded. Not everybody responded. And with itch, the patients who got better, their IL-31 went down, and the ones who didn't, they did not go down. So there are many translational studies that we've been able to do based on this trial. So where did we go with this? Well, it's now an ongoing global phase 3. They've had a pivot to doing more systemic DM, different outcome measures, a lot of regulatory issues, and certainly hard in a pandemic to do a trial, but it's a very important trial. So there's an opportunity to understand more about the variation in inflammatory cells and pathways in DM. Studies on itch and DM are really ongoing, and also the role of CB2 receptor as a regulator of inflammation in DM. So in summary, defining the disease is key. Validated outcome measures open doors to studies. The importance of basic translational clinical studies, I think, is obvious to all of us, but there's been really not so many done in autoimmune disease and translational and clinical. I have to emphasize that mentorship is critical, and it's important to have time, institutional, and funding support to advance the field, and collaborators are very important. And this is a time of immense opportunities. I have to acknowledge my funding sources, and also the many collaborators, students, fellows, statisticians, and coordinators that have been so important for the studies and the patients. Thank you very much.
Thank you so much, Dr. Wirth. That was just an incredibly enlightening discussion, as well as one that I hope will motivate more pediatric dermatologists to start doing research in our rheumatologic disorders. Ken Kramer's asking a question: have any of lupus or DM studies been carried out in children?
So actually, that's another area that I think is really ripe for you guys. The pediatric rheumatologists have some vocal representation on lupus foundations, and there's a lot of agendas that get set through the patient advocacy groups, and they are trying very hard to bring pediatric lupus into focus. And I think that to the extent that working with PeDRA and working with these organizations, you can really elevate the thought about skin as well, but there has not been all that many studies. It's been much harder to get pediatric lupus studies, but I think that that will change.
Quick question about, do you know the source of the IL-31 from Brian?
Brian. So, that's something that's ongoing that we're working on, and we are getting some answers, but it's a little early yet to say. But as I showed you, I know the myeloid DCs are making IL-31 for one. Yeah.
Great. Quick question from me. Intrigued by the ustekinumab story, because that is a drug that we know very well in dermatology and maybe could possibly get. Could you tell us a little bit more about what you think the mechanism is by which that might be useful? Does it selectively work more with that subgroup that has higher TNF levels, for example? You didn't really talk about the Th17 pathway or IL-22, IL-23. Just any quick comments?
Yeah, I mean, Th17 cells haven't been really found, at least in cutaneous lupus, to be that important. So I was a little surprised to see these results, actually. And I don't know that we know how it's working. And, I think it's still there. We need to learn more, basically. I think there must be some interaction with that pathway that we haven't previously thought about as much. Yeah.
Thank you. Well, I think at this point, we probably should go on. So maybe Carrie could start introducing our speakers.
Definitely.
Thank you so much.
Thank you again, Dr. Wirth. That was excellent.
My pleasure. And I'm going to stop sharing.
We are going to move to our first abstract. It's going to be presented by Madison Mack, formerly of WashU, now a captain of industry. And she'll be talking about NK cell deficiency in atopic dermatitis.
All right, thank you very much for the introduction and for the opportunity to speak at this session today. Can everyone hear me? Carrie, can you confirm? Okay, excellent.
Yep, if you want to, yep, slideshow, perfect.
Yeah, there we go. Okay, excellent. So today I'm going to give a little snapshot of work that I did in Brian Kim's lab at Washington University in St. Louis, and was actually recently published in Science Translational Medicine. And before I start, I should disclose that I am now a scientist at Sanofi, also working on type 2 inflammation, and I am a co-inventor on a patent related to this work. So most of you are likely familiar with atopic dermatitis, or AD. When we think of AD, we tend to think about the skin lesions, so that we think about cells like innate cells like ILC2s and eosinophils, as well as adaptive cells such as Th2 cells accumulating in lesional skin and driving pathologic type 2 inflammatory responses. However, we also know that AD is a systemic disease. It has elevated IgE, serum chemokines like TARC, eosinophilia, and a risk of increased risk of acquiring other allergic disorders such as food allergy and asthma. So this is also accompanied by a decrease in antiviral immunity, which is a little bit less well understood in AD, but it's evidenced by an increased rate of cutaneous warts, herpes, and molluscum for these patients. And this impaired antiviral immunity sparked our interest in NK cells, which are important antiviral effector cells, and have been shown to be altered in AD, but the impact of this on the disease was really unclear. So we started doing a retrospective analysis of 25 moderate to severe adult AD patients and a matched control cohort at Barnes-Jewish Hospital associated with WashU. And in this study, we found that there were similar levels or similar numbers of CD4 and CD8 T cells and B cells in the two groups. However, the AD patients had a significant reduction in their peripheral blood NK cells. And when we did an ROC, or receiver operating characteristic analysis of this low blood NK cells, we found that low blood NK cells were actually a fairly good diagnostic indicator of AD over our control all-comers cohort. So human NK cells, they're a diverse population of cells, and they express a wide array of both activating and inhibitory receptors. And this diversity has been beautifully studied and characterized by other groups in the field and subsetted into a number of different NK cell populations, which can be divided into CD56 bright cells, which are more proliferative and less cytotoxic, and CD56 dim cells, which come in several different maturation stages and are more cytotoxic and the most common type found in the blood. So in order to capture the combinatorial diversity of these cells, we turn to mass cytometry, also known as CyTOF. And for this study, we took PBMCs from AD and control patients and stained them with the 36 marker metal conjugated antibody panel and performed CyTOF on it. And then we used a dimensionality reduction approach called VISNI to visualize the data. And I'm going to summarize a lot of this analysis here in this heat map, which is showing you the 6 different subpopulations of NK cells here on the y-axis that we identified, and then the median expression of each of the 36 markers that we used in our CyTOF analysis. And so this is for all of the donors, and in order to understand whether there were any AD-specific changes in these populations, I performed a comparison of the frequency of each of these different subsets between the control cohort in blue and then the AD cohort in red. And what this showed is actually a selective reduction in the mature CD56 dim subset of NK cells in AD patients compared to the controls. And we were also able to look at 6 AD patients before and after treatment with the anti-IL-4 receptor alpha antibody dupilumab. These patients all had a nice clinical response to dupilumab after at least 3 months of treatment. And they also had a significant increase in their peripheral blood NK cells, which suggested that NK cells may be treatment responsive. So to try to understand why these patients were lacking this mature CD56 dim NK cell population, we sorted out their peripheral blood CD56 dim NK cells and performed RNA sequencing. And for the sake of time, I don't actually have time to go through all of the data for this, and I encourage you to check out our paper if you're interested. But in short, what we found is that the CD56 DIM and K cells from AD donors. They had an activated and a pro-apoptotic transcriptional profile. And this was correlated with an enhanced susceptibility to activation-induced cell death in vitro. And interestingly, these patients had elevated levels of cytokines like IL-12 and IL-18, which prime NK cells for activation-induced cell death. So now that we know that these patients have an NK cell deficiency, we wanted to see whether or not we could intervene on the pathogenesis of the disease or the pathogenic disease process by increasing their NK cell numbers. So to do this, we used an IL-15 superagonist, which is an IL-15 cytokine bound to the extracellular domain of IL-15 receptor alpha and fused to an FC fragment. And this compound has been used both in mice and in humans to induce NK cell proliferation very successfully. And so we use a mouse model of AD-like disease where we treat mice on the ear skin daily for 7 days with MC903 or calcipotriol. And this induces a robust AD-like inflammation. And then after starting to induce this inflammatory response, we treated mice with IL-15 superagonist or isotype. And as you can see, mice given the superagonist had an increase in both their blood and their skin NK cells. And surprisingly, they also had a reduction in pathogenic type 2 cells, such as ILC2s and eosinophils. And this resulted in a decreased overall clinical disease severity. So you can see here is a picture of isotype treated mice, and the superagonist treatment reduced both the scaling and the erythema we saw in the mice, and we quantified this using a clinical scoring system similar to the EASY. So taken together, these data suggest that boosting NK cells is able to ameliorate AD-like disease in mice. So in summary, in contrast to a lot of the current therapeutic approaches that are in development or available now that are trying to repress the type 2 immune response in these patients, perhaps an alternative approach would be to enhance an endogenous regulatory response by increasing NK cell numbers. So with that, I want to thank everyone in the Kim Lab and at WashU and all of our collaborators and our funding, and perhaps take some questions through the chat. Thank you.
Thank you very much. I don't see any questions, but I'm going to ask one. Could you tell us if you were able to boost the NK and you found the reduction, for example, in ILC2 cells and EOs? Did you look at the cytokine expression? Were you able to actually reduce the Th2 pathway and what we think of as drivers in tissue by doing that?
That's a great question, and we didn't actually look at cytokines either in the skin or in the serum, so that would be a really great follow-up to this.
So there was just a quick question whether the mouse EASY score was called MEASY. I don't know if you had that in there, but maybe a suggestion.
That's a great suggestion. Yeah, we'll consider that.
And then have you looked at the NK cell levels in the human skin of AD lesions? Are NK cells potentially migrating into skin and therefore decreasing? Is that what's leading to the depletion in blood?
Right, that's a great question. So NK cells in skin are pretty rare. In steady state, they're very rare, and we didn't do any cellular analysis of NK cells in AD skin, but on a transcriptional level, we can see that there is increased NK cell activity, which suggests that there may be more NK cells present. And I do think that there is likely some migration of NK cells from the blood into the skin, but the numbers don't quite add up in terms of being able to deplete patients of NK cells that significantly, if that makes sense.
Okay, well, thank you for the wonderful presentation, and we'll go on.
Thank you.
All right, Dr. Mack, that was fabulous. Next up, we have Aislinn Diaz. She's going to be talking with us about RNA-seq profiling of tape strips from infants with atopic dermatitis. Thanks so much for joining us, Aislinn.
Hi, good morning, everyone. Thank you so much for having me. Hi, Dr. Poller. How are you? Nice to see you. So let me just go ahead and share my screen. Can you hear me pretty good?
Perfect.
So I'll do this, share. And then, okay. So yes, I'll be discussing today RNA-seq profiling of tape strips from pediatric AD with atopic dermatitis that shows profound barrier and immune abnormalities. So for disclosures, I have none to disclose. So just a little bit of a background, atopic dermatitis is a chronic relapsing inflammatory skin disease. In fact, one of the most common inflammatory skin conditions in both children and adults. It typically begins with a defective skin barrier in which foreign antigens are able to penetrate. And then during the acute state, T helper 2 cells go ahead and induce interleukin 4 and interleukin 13, which act together through a common receptor to inhibit keratinocyte terminal differentiation, which ultimately disrupts the skin barrier. So T helper 2 cells also secrete an itch-inducing cytokine, interleukin 31, which leads to the itch scratch cycle and ultimately resulting in lichenification. Other T cell subpopulations such as T helper 1, T helper 17, and T helper 22 can also be detected in the skin as well. The interleukin 22 synergizes with interleukin 17 to go ahead and induce S100s, which also induces epidermal hyperplasia. And in a chronic state, T helper 1 cells predominate, producing T helper 1 products such as interferon gamma, CXCL9, CXCL10, and other cytokines that promote immune responses against intracellular pathogens. Let's see. So AD typically occurs in infancy and early childhood. A national survey of children's health reported a prevalence ranging from 8.7 to 18.1% for patients younger than the age of 17 years old in the US. And for these patients, most of them are younger than the age with disease onset occurring before the age of 5 in 85% of patients. Now, in the past, to understand the pathomechanisms involved in AD, skin biopsies have been instrumental in uncovering biomarkers that drive its pathogenesis. However, biopsies are invasive, causing scarring, pain, and a potential risk for infection, limiting their utility in large pediatric population. Now, in light of active therapeutic development for pediatric AD and longitudinal ongoing studies, there's a great need to develop a minimally invasive approach to investigate biomarkers. So the objective of our study was to identify a global transcriptomic AD profile in early pediatric AD using tape strips. Now, tape strips were previously used to differentiate pigmented lesions from melanoma, and in recent times, its use has been extended to inflammatory skin diseases. Tape stripping is a minimally invasive non-scarring approach utilizing serial adhesive films to capture the stratum corneum as well as the upper part of the granular layer. Now, in our study, we utilized 16 serially collected 16D squam tape strips from lesions in the antecubital fossa, from lesional and non-lesional skin, as well as healthy control skin. We also utilized RNA sequencing and Spearman correlations to evaluate the relationship between mRNA expression and clinical measures. Now, in our study cohort, we included 19 infants and toddlers between the ages of 0 to 5, with the majority being younger than the age of 1, with early onset, that being less than 6 months of disease initiation, and a severity of moderate to severe AD, with an average score rad of 56.5 and an EASI score of 21. For comparison, we utilized 17 healthy controls that were similar in age. Now here we have a heat map illustrating a transcriptome by RNA sequencing, and using a criteria of fold change greater than 2 and FTR less than 0.05, we were able to identify more than 2,000 differentially expressed genes and lesional and or non-lesional AD tape strip skin in comparison to healthy controls with 100% sample recovery. As you can see, the heat map illustrates a drastic difference in AD non-lesional and lesional skin in comparison to healthy controls, where blue is representative of downregulation of gene expression and red is representative of upregulation. Now, considering the main driving forces of AD pathogenesis, we first looked at immune response. We evaluated a previously reported immune gene subset demonstrating significant dysregulation of inflammatory genes in both lesional and non-lesional AD tape strip skin, as illustrated in this heat map. Now, some of these genes that were highlighted were some like innate immunity, such as interleukin 8, markers of the T helper 2 axis, such as interleukin 4 receptor, which is a target of dupilumab, as well as interleukin 13. Both of these are which are known key AD cytokines. We also noted markers of the T helper 17, T helper 22 axis, such as interleukin 22, interleukin 36G, CCL20, and S100s. Now, we also noted a lack of T helper 1 activation. Data isn't shown, which is actually in line to previous pediatric skin biopsy studies that highlight this T helper 2, T helper 17, T helper 22 skewing in pediatric AD tissues, and a lack of T helper 1 activation that typically characterizes AD adults with longstanding disease. Now, we next looked at barrier abnormalities. So the heat map to the right is illustrating nicely this downregulation of barrier-related measures in both non-lesional and lesional AD skin compared to controls, with even greater decreases noted in AD lesional skin. Now, some of these markers that were significantly downregulated across AD tissues included filaggrin, which is a marker of terminal differentiation, markers of lipids, which is FA2H, fatty acid 2 hydroxylase, fatty acid elongase ELOF3, as well as a downregulation of claudins. Next, to evaluate changes in various immune and barrier gene signatures, we performed gene set variation analysis of previously published gene sets. As you can see, T helper 2 and T helper 17 pathways were significantly enriched in both lesional and non-lesional AD. T helper 22 showed significant increases in non-lesional AD and trended towards significance in lesional compared to normal skin. However, no significant dysregulation was noted in the T helper 1 axis. And in regards to terminal differentiation and lipids, significant modulation was seen in lesional AD skin versus controls. Now, we also wanted to determine if these biomarkers correlated with clinical severity measures. And just to point out a few examples, for lesional skin, we found significant positive correlations between body surface area and markers of T helper 17, such as interleukin 17F, interleukin 36G. And for transepidermal water loss, which is an objective functional measure of the barrier, we found significant negative correlations with filaggrin 2 and filaggrin, which are markers of terminal differentiation in both lesional and non-lesional AD skin. Now, in summary, our data demonstrated that the AD phenotype of early onset PED AD can be accurately captured using tape strips. We were able to obtain 100% recovery rate across all tape strip samples and able to identify more than 2,000 differentially expressed genes in lesional and or non-lesional AD versus controls. Now, using RNA sequencing, our results showed a global tape strip profile that was characterized by significant increases of key immune cytokine pathways such as T helper 2, T helper 17, and T helper 22, as well as dysregulation of barrier-related measures previously described in biopsy studies. Now, lastly, we also noted significant correlations between clinical measures and immune and barrier mRNAs in lesional and non-lesional AD skin. Now, overall, these findings support that tape strips are a minimally invasive objective approach to track cutaneous disease severity to predict disease course, comorbidities, and therapeutic responses. And with that, I would like to conclude and say a huge thank you to my PI, my mentor, Dr. Gutman-Yaski at the Icahn School of Medicine and her team, Dr. Amy Powell and her team from Northwestern of the Feinberg School of Medicine, as well as Dr. James Kruger and his team at the Rockefeller University. So thank you.
Thank you very much, Arlene. That was beautifully presented. And we have a question for you here. Actually, a few more coming in. First question is, can you tell us whether there's been any study of this over time in the same person? Would it modulate with time and be responsive to change? And also whether you did multiple on the same individual. Clearly, you did that on the same individual with non-lesional versus lesional, but if you did it in other areas of the body, for example, that were affected by lesions.
Yeah, so to my knowledge, all the samples were created in the antecubital fossa or anywhere near where the lesions were present, and they were collected for the same individual. So for the 16 tape strips that were serially collected, 3 to 16 were pulled together, and that's what was used for the tape strips. And then the first question, what was it again, Dr. Pollard?
Responsiveness. Over time, does it change? And I don't think you have that either.
Yeah, so I think that's the idea with using tape strips right now. I know there's a lot of ongoing longitudinal studies going on, and hopefully in due time, we'll be able to get a better response if we're actually are seeing changes from children at such a young age, and if we're getting any differences in adolescence as well.
there was also the question of whether the tape stripping itself could be altering the pattern. It's kind of hard to know that, isn't it?
Yeah, so I think for the use, do see the point. I think that's actually one of also the limitations when you're comparing AD skin that's already altered in comparison to normal skin in which the full barrier isn't impacted. And I know right now there are studies that are showing the differences in a whole skin biopsy versus tape strip skin on the same exact sample. And they're actually showing that there's not really much of a grade difference in that sense.
Two other quick questions. What kind of sequencing library did you use? What was the sequencing depth that was achieved? And also a question, is there variation in the amount of RNA recovery among different strips from the same person? If you could just answer both of those, and then I think we'll move on.
Yes, so we use the RNA AmpliSeq libraries that were constructed with the Ion AmpliSeq Transcriptome Human Gene Expression Kit. In terms of, I believe it was the sequencing that was asked, I'm not sure.
Depth.
Yeah, I'm not sure how much was used, but per sample that we utilized, it was 5 nanograms of RNA.
Okay, the recovery among different strips from the same person, you said you collected 16. Did you look at RNA recovery strip by strip at all?
No, I personally didn't look at it, but our technician, I'm sure, has that data on file.
So that was from Ken Kramer. Maybe we could get back to him later.
Yeah.
All right, and we'll move on. Thank you so much.
Great, excellent. Thanks so much, Aisleen. Next, we have Dr. John DiGiovanna. He's coming to us from NIH NCI, the Center for Cancer Research, going to be talking about trichothiodystrophy.
Hi there all. Let's see if I can put this where it's supposed to be. And get a pointer. Thanks for the invitation. I really enjoyed putting this together and for the opportunity to present some of our work. I'm going to be talking about trichothiodystrophy, a multi-system disorder with early onset debilitating hip degeneration. So trichothiodystrophy, or TTD, is an autosomal recessive disorder. It's quite rare. It's one of the hereditary disorders of genome instability, along with xeroderma pigmentosum and Cockayne syndrome. And in this entity, there are molecular abnormalities, both of DNA repair, but also of transcription.
John, do you know that your slides aren't up?
Oh, no. Sorry.
Share screen.
Okay. Let's see if I can get this correct. Are you shared?
Not yet. Oh, here we go.
All righty.
Now we're here. Go ahead.
Sorry about that. So TTD is a rare disorder of genome instability. This is a young boy with trichothiodystrophy. You see the smiling, outgoing face. Sorry, I'm not actually.
Just put in presentation mode and we'll be set.
Yeah, it doesn't seem to want to do that.
So you're going down to the bottom there, yeah. Overall, there you go. That should do it.
There we go. Okay, great. Sorry about that. It's a little bit different doing than this when you have somebody else do all this for you. So again, this is a disorder of both DNA and DNA repair and transcription. The patients have brittle hair with low sulfur content. When you examine it under polarized microscopy, you see this characteristic tiger tail banding. And the patients have clinical features that are representative both of abnormal DNA repair, but also of abnormal development. And it's caused by a variety of different genes, some of which have been related to abnormal transcription, but others are of unknown function. So of the patients that we followed, we have a cohort of 38 patients. Most of these, 23, are in the XPD complementation group. Difference between males and females is approximately equal. However, the disease has a high mortality. So 26 of the patients have died, and they die at a very young age, an average age of 10. Within our group, the patients have a high frequency of the features of different organ systems that have been described in the literature: the hair, the skin, musculoskeletal system. Mothers carrying affected babies have bad pregnancies, developmental delay, hematologic, ophthalmologic, and a variety of abnormalities. So we've begun to recognize that TTD patients have a number of developmental abnormalities. Carrying affecteds have troubled pregnancies with preeclampsia or HELLP syndrome, hemolysis, elevated liver enzymes, and low platelets, which has a mortality to it. Patients are often born with a very mild collodion membrane, which quickly improves and leaves a mild ichthyosis in childhood. These two unrelated boys have a similar habitus. They're both 10 years of age, and they both have short stature. Micronathia is another developmental abnormality. They don't lay down myelin in the brain, as shown on the MRI. And they also have this distinctive bone abnormality of the central skeleton being dense, having osteosclerosis, and the peripheral skeleton having osteopenia. So we began to see children like this 7-year-old girl who was able to walk with a walker, and she needed the walker because of ataxia until she was 6. At that point, her hip x-rays were normal. At age 7, she developed difficulty walking because of hip pain. An x-ray of the right hip showed an effusion in bilateral coxavalga, malposition of the leg. At 7 years, 8 months, an MRI showed osteonecrosis of the right femoral epiphysis. At that time, her left hip was normal on imaging. A few months later, she was unable to walk at all. Physical medicine evaluation at 8 years of age showed that she sat in a W-sitting position. She had bilateral leg weakness, hamstring tightness, limitation of range of motion of the hip, and she was scheduled for surgery where she had bilateral adductor tenotomies and tightening of the hip tendons. 2 months later, she had further deterioration of the right hip on imaging. She'd been unable to walk for the year post-surgery, and at 9 years of age, she had a similar imaging deformity of the left hip with subluxation and osteonecrosis or avascular necrosis. She died 3 months later of MRSA pneumonia, also with associated low serum IgGs. And so this was 2 years after the onset of walking difficulty related to hip pain. So here's a series of imaging from this child. At 6 years of age and by early 7, she had normal epiphyses. She began to develop difficulty of the right hip. This progressed. She had necrosis, AVN of the right hip, and then developed it bilaterally. This shows a 3D rendering, not of this patient, but of another patient derived from a CT scan who had similar difficulty with walking. And you see a visual representation of the destruction of the femoral neck and the femoral head. On one patient, we were able to actually obtain bone histology, and the histology of the femoral head shows the osteonecrosis with dense cortical bone. And on a higher microscopy, you can see that the lacunae are empty of osteocytes, and the bone pathologist said this is what AVN, avascular necrosis, would look like, but this was after it had occurred. Who gets these within the subgroup? We have a representation of many of the different genes underlying TTD. It seems that the 9 patients who had this debilitating hip degeneration all were in the XPD genotype. Excuse me. They had onset of hip pain at an average age of 8, onset of the imaging findings by 9, and of the 6 patients who had it bilaterally, this occurred within another year by age 10. We had a number of other features occurring within our cohort of musculoskeletal abnormalities. Several of the patients had a coxavalga or turning out of the leg, the central osteosclerosis, peripheral osteopenia, this W-sitting and contractures at the hip joint. Perhaps a better way of looking at the data is with this array that shows the different mutated genes in each patient, the presence of this debilitating hip degeneration, either avascular necrosis or something radiographically similar to it, coxavalga, osteosclerosis, osteopenia, the W-sitting, and lower limb contractions. What you can see is that 9 of the 36 patients within the XPD group, 39% had this debilitating hip degeneration. None of the patients in the other complementation groups developed this problem. So how could this happen? Well, Donata Orioli is a basic scientist in Pavia, Italy, who we've worked with, who studies transcriptional abnormalities in TTD. And she's shown a variety of abnormalities in collagen homeostasis. The TTD primary dermal fibroblasts seem to have low amounts of collagen 6 alpha-1 subtype. And it appears that it occurs because of inability of the mutated TF2H complexes. TF2H is a basal transcription factor, and it's unable to remove this regulatory element, the sterol regulatory element binding protein, from the collagen 6A1 promoter. And that is what's necessary to sustain a subsequent high rate of collagen 6A1 transcription. And they've termed this transcription derepression. In addition, there are other abnormalities. She showed overexpression of matrix metalloprotease, MMP1, in TTD fibroblasts from patients who have the mutated XPD compared to their healthy parents. And MMP1 degrades interstitial collagens of the extracellular matrix. So because of this, we did a survey of the literature. Sandra Follerin, who's a postback in our group, trying to see what other diseases of the epiphyses or hip degeneration are related to collagen abnormalities. And there are a number of these that have been described. These are germline mutations in collagen that have been associated with similar clinical phenotypes. And we hope to pursue this to try to better understand this abnormality. In summary, TTD is characterized by short, brittle, sulfur-deficient hair. As you know, tiger tail banding and polarization is quite characteristic. But the patients have a wide spectrum of developmental abnormalities. Musculoskeletal abnormalities are common. They can have severe morbidity. Most of the kids become unable to walk, and they're poorly understood. The TTD abnormalities in transcription have been associated with collagen abnormalities, and we wonder whether or not this could be related to the pathophysiology. And characterization of these TTD development abnormalities may help us understand normal musculoskeletal development in a better way. So thanks for your attention. I'm happy to try to answer any questions.
And thank you so much for that lovely talk. We are actually running about 10 minutes behind, and we don't have any questions up, so I'm just going to move on to the next speaker, and thank you again.
Yep, that was great. I love that we're able to see kind of a broad array of pediatric dermatology topics here, and the basic as well as clinical correlations has been fabulous. Our next speaker is going to be soon to be Dr. Joshua Hekmacha. He is coming to us from Western Michigan and USC Keck School of Medicine. He's going to be talking with us about acne patients on isotretinoin and their psychological features.
Hello, everybody. Can everybody hear me?
Yep, that's great.
Perfect. Let me share my screen. Can everybody see the slides?
Yep, looks good.
Perfect. Good morning, everybody. My name is Joshua Hekmacha, and I'm a medical student at the Western Michigan University Homer Stryker School of Medicine, and I'm currently doing a research year under Dr. April Armstrong at the University of Southern California. Thank you for inviting me to present my research titled Acne Patients on Isotretinoin Experience Less Psychological Distress and Depression Symptoms as compared to those on oral antibiotics. I have no conflicts of interest. Dr. Armstrong conducts clinical trials and her disclosures are listed. None are relevant to the medications in this talk. Acne is one of the most commonly encountered diseases in dermatology, affecting 50 million people in the United States. Because acne commonly affects the face, acne patients experience higher rates of depression and psychological distress. However, little is known regarding the differential effects of systemic anti-acne treatments on mental health. This study's aim was to determine whether differences exist in mental health outcomes between patients treated with isotretinoin versus oral tetracyclines. Tetracyclines included in this study were doxycycline, minocycline, and tetracycline. We performed a cross-sectional study using nationally representative data from the 2004 to 2017 Medical Expenditure Panel Survey, also known as MEPS. The study population was acne patients who were treated with isotretinoin or oral tetracyclines during this period, who also completed questionnaires on their mental health outcomes. These included depression symptoms as measured by the Patient Health Questionnaire 2, and psychological distress as measured by the Kessler 6-item psychological distress scale. The first measure of mental health was the Patient Health Questionnaire 2, also known as the PHQ-2, a validated 2-item screening and monitoring tool for depression. It consists of 2 questions, which are as follows. Over the past 2 weeks, how often have you been bothered by any of the following problems? 1, little interest or pleasure in doing things, and 2, feeling down, depressed, or hopeless. Lower scores represent fewer depression symptoms. The questions are scored on a 4-point Likert scale from 0, not at all, to 3, nearly every day. A score of 3 or more is considered a positive screen for a depressive disorder. The second measure of mental health was the Kessler 6-item psychological distress scale, also known as the K6, a validated 6-item measure of psychological distress. It consists of 6 questions. During the past 30 days, how often did you feel? 1, nervous. 2, hopeless. 3, restless or fidgety. 4, so depressed that nothing could cheer you up. 5, that everything was an effort. And 6, worthless. The questions are scored on a 5-point Likert scale from 0, none of the time, to 4, all of the time, with lower scores representing less psychological distress. Adding scores across each item creates a global score from 0 to 24. Scores of 0 to less than 5 are usually considered to mean no or mild psychological distress. Scores of 5 to less than 13 are usually considered to mean moderate psychological distress, and scores of 13 to 24 are usually considered to mean serious psychological distress. We performed a multivariable linear regression adjusting for multiple covariates such as age, gender, race, ethnicity, and insurance coverage. Our results showed that the mean PHQ-2 score from patients on isotretinoin was 0.28, which was significantly lower than those on oral tetracyclines. That is, patients on isotretinoin had fewer depression symptoms than those on oral tetracyclines. The mean K6 score from patients on isotretinoin was 2.494, which was also significantly lower than those on oral tetracyclines. That is, patients on isotretinoin experienced less psychological distress than those on oral tetracyclines. After adjusting for sociodemographic and clinical characteristics, the adjusted linear regression showed that isotretinoin was associated with fewer depression symptoms and less psychological distress. Our results showed that isotretinoin was associated with less psychological distress and fewer depression symptoms as compared to oral tetracyclines in acne patients. These results contrast prior studies reporting an association between isotretinoin and depression. Rather, this study suggests that acne itself may be an independent risk factor for depression. Furthermore, these results also corroborate and extend recent findings that isotretinoin is associated with improved mental health outcomes. Thank you for listening. I would like to also thank Dr. Armstrong for everything she has taught me during this year and my research team, which is pictured here. Thank you very much.
Thank you so much. That was really nice to hear a little bit about how depression is alleviated by isotretinoin. But could you tell me a little bit about when in the cycle of isotretinoin or after starting these antibiotics you did this study? Did you have a particular time? Because we all know that people who are getting better, for example, might feel better. And if they're not, they might not. Did you correlate that at all with the severity at the time and the duration on the medication?
Because we pooled the data, it was hard to tell at which time when they started the medication, but that's a very interesting question, and it could be good for future research to see if at different time points of treatment there were different mental health outcome measures.
Okay, thank you very much.
Thank you.
All right, and then our next abstract will be presented by Cherie Wright. She's coming to us from Hofstra and Northwell Health, and she'll be discussing incidence of depression among patients with hidradenitis.
Good afternoon, everyone. Just pull this up. Can you all see my screen?
Yep, if you go into that presenter mode, you'll be good to go.
Perfect.
Great.
Great. Yes, so good afternoon, everyone. My name is Shari Wright, and I'm a research fellow in the Dermatology department at the Zucker School of Medicine. Today, I'll be presenting our data on new onset depression among children and adolescents with hidradenitis suppurativa. I have no conflicts of interest to disclose. Dr. Garg, my PI, has the following disclosures. None of the relationships listed had any input to this study. So for a little bit of background, hidradenitis suppurativa is a chronic inflammatory disease involving the follicular unit, which results in painful nodules, draining abscesses, and the formation of fistulas, sinus tracts, and scarring. It most commonly affects the inframammary region, the groin, perineum, and axilla. The physical symptoms associated with HS are often debilitating and can lead to significant psychosocial impairment and decreased health and skin-specific quality of life. However, the risk of new onset depression in the pediatric HS population is not well established. So we sought to compare the incidence of depression in pediatric HS patients and control patients. The study was a retrospective cohort analysis using the Explorers database by IBM Watson. Explorers is an electronic health records database of 56 million people across the country, which represents roughly 17% of the US population. The analysis was limited to patients ages 17 years or younger, and we had two main study groups: HS patients who were identified by at least one ICD code for HS, and control patients who had no diagnosis of HS ever. We match HS patients and controls based on zip code and index year, and we excluded patients with a history of depression prior to HS diagnosis. Our primary outcome of interest was an incident depression diagnosis. We adjusted for demographics, number of healthcare encounters, smoking status, alcohol use disorder, substance use disorder, and BMI. So in terms of our results, there were roughly 3,000 HS patients and roughly 12,000 control patients in this analysis. HS patients were more likely to be older, female, African American, and obese. HS patients, approximately 9% of the HS cohorts were smokers compared to 4% of our control patients. The crude incidence rate of depression was 4.2 per 100 person years among HS patients compared to 2.3 per 100 person years for control patients. In unadjusted analysis, HS patients had an 87% increase in the risk of depression, and after adjusting for demographic and clinical covariates, HS patients had a 26% increase in the risk of depression. We were also interested in determining which clinical characteristics were most closely associated with new onset depression. We found that female sex, Caucasian race, obesity, and smoking status were all associated with new onset depression among pediatric HS patients. So to put this into context, female patients with HS were 2.13 times more likely to develop depression compared to male patients with HS. Our study had several strengths and limitations. We were able to utilize a large, diverse patient cohort and adjust for a number of demographic and clinical covariates. However, we could not capture patients with HS who did not seek care in the health systems included in our database. Despite these limitations, our data suggests that HS is associated with an increased risk of depression. This increased risk is independent of other common risk factors for depression. Characteristics such as obesity and smoking status contribute significantly to the development of depression within pediatric HS patients. And this is very similar to what we see in the general population. Given all of this, providers may consider periodic screening for depression among HS patients, especially among those who have additional depression risk factors. I'd just like to take this time to thank PeDRA for inviting me to share our data at this session with you all. And I would also like to acknowledge my mentors, Andrew Strunk, who is our biostatistician, and my PI, Dr. Amit Garg. Thank you, guys.
Thank you very much. We have a question here about whether you looked at passive tobacco, which I think means in the environment. So children might have a parent who smokes in presence, Was that something that you asked about?
So unfortunately, the database does not provide such information. So we were only able to gather smoking status as an ever, never type of variable. But that would be definitely something that would be useful, since most of the pediatric population would have more secondhand exposure as opposed to primary exposure themselves.
And do you know whether the females in your study were more likely to seek treatment for the depression than males across this disease, or did you look across others?
That's also a very interesting question. We did not look at whether or not they did look into treatment for the depression.
Okay, well, thank you so much. I think we better close the session now.
Thank
Yes,
you,
so
guys.
thank you all for attending so much. This has been a great session in terms of variety of conditions that we've covered and some great questions from the audience. We really appreciate the attendance and are happy to close. Thanks so much.